Well, hello everyone, and welcome to Crop Hour. This is day 3 or the final day of our sunflower and pulse week. My name's Patrick Wagner. I'm the entomology field specialist based out in Rapid City. We got Chris Graham on the line today. He's going to be talking to us about spring and winter pea production here in South Dakota. I want to mention to everyone that if you're on here to get your CCA credits, I will be putting up a QR code at the end of the day here or the end of the hour, and you can scan that QR code and get your CCA credits that way. So Chris, do you want to go ahead and share your screen?
Yep.
So Dr. Chris Graham, he's an assistant professor and extension agronomist here at SDSU. He's also the manager of the West River Research Farm out in Sturgis. So yeah, tell us all about pea production here, Chris. Take it away.
Okay. Thanks, Pat. Is that looking all right?
Yep, looks good to me.
Okay. So before I get started, it looks like we're a small enough group that if there's questions or discussion, certainly we could go ahead and interrupt at any time. So Pat, if something comes up in the chat box, please feel free to jump in. And we can talk about specific issues that people are having too. Like I said, it's a group that's small enough that we can handle that pretty well, I think. Anyway, so as Pat said, I manage the West River Research Farm out in Sturgis, just east of Sturgis. That picture here that you can see is looking from the farm, and in the top left you can see there's Bear Butte. So that's kind of gives if you're familiar with Western South Dakota, that kind of gives a sense for where we're located. By background, I'm a soil scientist, and I do a lot of work with various microbes and pertaining to soil health in particular. And so I'll talk a little bit about some of our work with one microbe in particular called rhizobia later on in the talk. But in general, this talk is geared towards newer producers. I know we have a pretty wide diversity of pulse growers across the state. We're still pretty small in terms of acres, but there's some farmers that have been growing them for many years. But generally what we're doing is always trying to expand the pulse acres. And so we're speaking to a lot of newer audiences or people that are unfamiliar with these. So that's kind of the idea behind this talk. But I said, if anyone wants to bring up any more advanced issues or anything else, feel free to put that in the chat and we'll address that. So just a little bit of background, where do the peas go? And what are they grown for? Basically, there are two market classes. There's green and yellow, and that just refers to the cotyledons within the plant. And there's not a lot of difference. They do have some different pathways in terms of marketing. By far, I think the yellow peas are produced more throughout the country and into Canada. But greens are, they're more or less the same. Greens are a little more finicky because there's a grading issue in terms of how the green color, and there's this bleaching that we tend to get where you don't have to deal with that in yellow. Most of the peas that we grow are exported or fed. That picture here in the left-hand corner is a picture I took when I was in Ethiopia a few years back, and it was a large warehouse it was just food aid. And so they had these tons and tons, literally, of these bags. You can see here split yellow peas. And so we send it off as food aid that can be made into a variety of edible products. But lately, the bigger or the hottest market, I guess, for these has been in the fractionation market, where the protein, these are relatively high protein, which we'll talk about. The protein is fractionated out and it's supplemented into other snacks, as these protein fortified snacks. And you'll see that if you look at the ingredients, it pops up all over the place. In this picture in the bottom left, we have this, it's actually a pea milk. I tried the chocolate milk. It was pretty good. But yeah, so if you look at these things, you know, everywhere throughout the grocery store, you see that now that there's protein in whatever the food item is. And there's a couple different protein sources that are used, but peas are certainly one of the major protein sources for that. And then, as I said, it also makes a good feed and forage for a lot of livestock producers. So this is a map. This is a few years old now, but this kind of gives a sense for where peas are grown. And I should clarify. So, you know, I say pulses. That's kind of a synonymous with legumes in general, but the pulses that we're talking about typically are chickpeas, lentils, and field peas. I'm focusing on field peas today because they're kind of a good entry level into the growing pulses. But if that's confusing, sometimes they are used interchangeably between legumes and pulses. At any rate, so this map shows where most of the production is concentrated. As you can see, it's mostly that northern tier of the high plains in the United States. And of course, it doesn't stop at the border. There's significant growth in Canada, more than the United States. But it's slowly migrating south. And you can see here in South Dakota, there's especially in the central part of the state there's some pulses grown. And it's going farther south every year. So for the spring peas, you know, they basically mimic spring wheat. And I always say if you're familiar with spring wheat, it's pretty easy to grow spring peas because the season very much mimics that in terms of planting time, harvest time. So a lot of the operations are quite similar. You know, you plant in our country early April, mid-April, and then harvest in mid to late July. So these are a cool season crop, and they'll germinate above 40 degrees. One of the issues that we have, or we find we have a lot of issues with, is the slow early growth of pulse crops, especially peas. So they'll germinate at 40 degrees, but the issue is that it might take, you know, 10 to 14 days, even 20 days, depending on how cool it is, before it actually emerges from the ground and starts to really grow. And so I think from an early or new producer, one of the biggest problems that typically occurs is that weed control is often quite difficult in those early, you know, because you have, say, a plant like kochia or sometimes brome, cheatgrass, that can germinate in the late fall or early winter, or excuse me, late winter, early spring, and it gets going, and so early pea growth does not compete well with many weeds. But one of the nice things about field peas is that they're hypogeal, as opposed to a lot of other crops that are epigeal. Hypogeal meaning that the growth point stays below the ground, and so what that means functionally is that if you get later frost, say in late April or early May, typically peas don't have any issue with it because that growing point is below the ground. It's insulated by the soil and it can tolerate that frost quite well. So looking at a couple different graphs here. So I started talking a little bit about green peas versus yellow peas. So one of the first questions is, well, how do they yield? How do they yield comparatively? And so this first box plot up here kind of shows a lot of data that we've collected over the years. And the box plot, which I'll show probably quite a few of those, just for those that are not familiar with this type of graph, basically what the box means is the top of the box and the bottom of the box are the 25th and 75th percentile. These whiskers here, sometimes they call it a box and whiskers plot. This is the range. So this is the minimum at the bottom of the range and the maximum up here for your confidence intervals. And then this thick bar right here is the average. So what this graph is showing is on the y-axis, you have the yield in bushels per acre, and then you have the green peas versus yellow peas. And again, this is over many, many years. And so there's a big distribution in the data. But basically, if you look at these averages, yellow peas out yield green peas by a few bushels in general. That's the only major difference that we see. I would guess it's probably 3 to 4 bushels on average per year. And there's going to be, if you see the spread and all these are each individual data points. And so there's going to be different varietal differences within these market classes, of course, as well. So protein, they're roughly the same. This is a couple percent, this is percent protein on the y-axis. But they're 20, somewhere between 26 and 27% protein on average. So that doesn't vary so much. Certainly wouldn't be statistical. Planting. We typically, so these are large seeds, most of you have seen peas somewhere. These are a much larger seed than wheat, which means that to germinate, they imbibe a lot more water. And they could, maybe 50 to 150% of their weight by mass of water is germinated or imbibed to germinate. So they take a lot of water to start growing. So we typically recommend a slightly deeper depth than wheat. We say, 1.5 for wheat, but this you say 2 inches or so. 2.5 might be a little extreme. But 2 inches is generally what we recommend. Target seeding rate was 350,000 seeds per acre. So if you do the math, if you figure maybe 2,000 seeds in a pound, there's a lot of pounds of seed that you're hauling around to plant these things. So it's bulky. We've done a lot of work on seeding rates. 350,000 was just the standard. It's always been the standard. It's a fact. It's the standard across most of the Midwest. And so these box plots on the top here, this is a single variety at all these different seeding rates, 250,000 all the way up to 400,000. Just a bit. And so this is the yield at each of those different seeding rates. And you can see this, again, this dark bar is the average. And you get a slight increase as you increase from 250 to 300,000, perhaps a couple bushels. And then it kind of levels off, and then maybe it even tails off a little bit as you get into these really high populations. And so essentially the take home here is that, 350,000 is pretty good. In general, you'd say, well, 300,000 is the same yield. Why would I add 50,000 more? The one thing I do kind of caution against, and I talked about this already, is the slow early growth. And so increasing seeding rates, I think, do help in some cases, especially in organic situations where you want that canopy to close as quickly as possible to start crowding out some of those weeds. So, so maybe you don't in general get this big yield bump going from 300K to 350K. I do like the idea that you have more plants out there to crowd out other weeds and to compete better with those weeds. Protein, no difference. So these kind of bounce around a little bit, but relatively they're pretty stable within 1% or 2, or within 1/10 or so percent. So it doesn't really affect the protein at whatever seeding rate. Timing is really critical with peas for when you plant them. So as I said, we target an early to mid-April. And so I'm just showing 1 year, and the data are pretty similar. We've done a lot of years of this. But what this graph is showing is these are a whole bunch of different varieties of peas here on the x-axis. This is the yield on the y-axis, and then this blue is the yield when they were planted on April 23rd. And then this orange color is a June 3rd planting. So this is really late. But and you can see there's this huge reduction regardless of variety, there's a great big reduction, roughly 65% in this case, across those varieties. And some varieties respond a bit more than others, like Carver. It's high, it remained high, even though it was, still pretty significantly reduced, whereas Shamrock had a much greater response to that later planting date. So in general, I think right now, I think it was extended last year, but the final planting date for insurance is May 5th. But we see these reductions even in the middle of May. We'll see 50-60% reductions by delaying planting. And I'll talk a little bit more about what that means for nitrogen fixation. But really, the physiologically, what happens here is that the flowering date is pushed out into, this is pretty extreme in this case, but the flowering date, the farther it's pushed out into June, or even early July, that's where we really start to run into problems with seed set and heat. And this table really kind of shows, I'll come back to what this part here, but this table kind of shows that what heat does to flowering on the flowering period in plants. And so they're showing the seed set. So that's the pollination period. That's when the flowers are pollinized, and then we start getting seed growth. And they have pod length, they have number of seeds per pod, and then the seed ovule. So that's like the number of potential seeds versus the actual seeds that were produced. And as you increase, you get up 91 degrees at flowering, it's not really affected. But once you start getting much over that, those really high heat days in the middle of summer, then you have a lot of things that are dramatically, pod length is decreased, the number of seeds per pod was decreased, and then of course the actual rate of seed set is decreased quite significantly. So flowering begins 40 to 50 days after emergence. So go ahead and back off or add another, let's say 10 days to that from planting to flowering, and you can kind of do the math on that in terms of planting dates. And this shows, going back to how long it takes for these to emerge and get growing, and at different temperatures, 38 to 45 degrees. Like I said, 40 degrees, it'll germinate. But you're looking at 3 weeks potentially to emergence versus 2 weeks and reduced as temperatures increase. And so there you are, kind of splitting the needle a little bit in terms of temperatures that are warm enough to germinate the seed and get it going quickly, as opposed to waiting too long for those soils to warm up. And then, you're again pushing that critical anthesis period. And then temperatures above 95 degrees or so really start to affect pollination period. So we do a lot of work funded by the South Dakota Pulse Council, looking at varieties across the state and running these variety trials. And so what I'm showing, this is from last year, 2022. We had 3 sites. So one in Pierre, one in Wall, and then one at the research farm in Sturgis. And typically, this is pretty standard in terms of sites. We have 3 to 5 sites every year. And we have 40 to 50 varieties that we'll test. And these come from commercial, seed companies, and then they come from breeders. There's pretty active breeders in North Dakota, Montana, Washington State, and Idaho. And so they'll submit what they think is going to be a good fit for our region. We don't solicit much beyond that. It just says, if we basically just do a call and everyone sends in the varieties that they think might be useful for growers there. So I put this in here. This is last year, if you remember, it was kind of a rough year for us in the West, much better in the central part of the state. But this is also the value of spreading out our sites, because seems like hail, we lose some sites every year to hail. Last year it was Wall, but great differences between Dakota Lakes, which is the Pierre site, and Sturgis, 13 versus 40 bushels. And this is just a kind of an example of what this looks like. This is posted on this website here at the bottom. It's at the, if you just Google SDSU field pea variety trials or something, it'll pull that up. But we generally publish the yield at each site, and then we publish the protein content. Right now, protein is not, it's not like wheat where you have a protein market where you might get a premium or you might get a discount depending on your protein content. I think it could potentially develop that way, because you can see there is quite a disparity in the range of protein. For example, Carver is always one of our best peas, but does really well across the state. But then it's always probably the bottom 2 or 3 in protein. So if you were looking at yield versus protein yield, grain yield versus protein yield, different varieties might rise to the top. So we publish this and we kind of show which varieties are tend to do better in different regions. Typically, we'll have a multi-year variety or averages as well. That's just showing one single season. So a lot of our pea acres are centered in the middle of the state. And so I just threw these out here. These are the ones that over a 3 year 3 years of testing are better performing ones. These happen to be all yellow peas. And so I talked about Carver a little bit, but they all have some different qualities, and different genetic resistance to certain diseases. In general, our disease is quite low. The thing that we might deal with occasionally is powdery mildew. But most often that's not a primary concern, at least for yield. Typically what happens is we get powdery mildew and then it kind of heats up and it kind of grows out of a lot of that stuff. But a lot of the varieties that are released for this area are resistant to powdery mildew. Now, talking a little bit about winter pea yield. So winter peas are quite new. Well, they're not new necessarily, but they're new to growers. And to my knowledge, we don't really have any winter peas being grown across the state. And I think the biggest hurdle has been the development of varieties that are hardy enough that can survive the winters. And so we started a couple years back just evaluating them, because I know there was a big push for these winter varieties. And so breeders started to look at, going back through some of the germplasm to develop some new ones. And so they've released a lot, and there's a lot in development. And, the obvious benefit is, I talked about spring peas are similar to spring wheat. Well, winter peas are very similar to winter wheat, and we can plant them in winter and, say September, and then they'll come off a little bit earlier. I talked a lot about the critical flowering period and the heat, so we can back that off a little bit into the earlier summer. And so there's a lot of potential value to adding winter peas in the rotation. Of course, the big question as well, is it going to survive our cold winters? And so here's some data that we put in 2021. We actually started in 2019. Unfortunately, we lost our 2022 trial due to some spray drift, but these are data from 2021. And so this is showing the yield this is kind of a switched around bar chart. So this is yield on the x-axis here, and these are different varieties. The ones that are actually named, like Vale, Keystone, Golden, and Blaze, are those that are released. So those are available for farmers to buy and grow. And these numbered ones are all experimentals. And so I think one of these was maybe released as I think it's this W2 that was released as well as a commercial variety by Meridian Seed. Anyway, so this is 2021 if you remember, that was another really dry year. In fact, it was worse than 2022. But these are the averages, these dots, this is the range that we saw here, but these are the averages in these different dots. And so, it's not great, 20 bushels. Our spring peas were 24 bushels an acre, so they were a bit lower. But the first take home here is, yes, we know that they can survive. 2021, actually the winter was wide open. There wasn't a lot of snow, not a lot of insulation. It was fairly dry, and we got some cold periods. So I think, we're still testing this, and I don't want to get overly confident on their winter hardiness, but I think, our winters in general, especially to the west where we're a bit warmer, they'll survive our winters. And so one of the hardest things that we came across with this is weed control. Organic growers, I think it's going to be interesting to try to do this, because you get a lot of, in theory, you can take care of some of your fall germinating weeds, but the later stuff, for us at Sturgis, prickly lettuce was a big problem, and it comes a little bit later in the growing season. And so it created a lot of issues for us. And I think that was, it's not the hybrid vigor of these different ones, or, if it was a hybrid, it's not the vigor of the plants. It's really just the cultural techniques and how we can manage these weeds. It's a bit more tricky, and we're still trying to wrap our brains around it. But protein, that was another issue. Tend to see higher protein, but that might be a little bit because smaller yields. But in general, I guess my point is that these can compete with spring peas. And I have a couple slides here just from other plains regions that I think have similar winters to us, that are looking at these as well. And so this is out of Colorado, and I think Fort Collins from these sites is most similar to us. And this is the winter peas that they had. They have it in pounds per acre. They have irrigated and dry land. And then these are the varieties that they have. So I should make a distinction, because often people think, well, winter peas, aren't those Austrian winter peas? And that's not the case. Those are a different beast altogether. They're similar, but they're not the same. And so they're much smaller seed. So these are same as the spring peas, the yellow and the green. And these are a couple of those were in our trials, but these are others that are released now. You can see, obviously, a big response to the irrigated winter peas, but generally, they produce seed, they produce yield, and so they survived, and generally pretty good. So it's a 60 bushel crop, and so if you divided that by 60, you would get your bushels per acre there. And this is just a comparison. So if you look, they don't have the average, unfortunately, but let's just say it's somewhere between the two of these. It's somewhere around 12, maybe 1,500 pounds on average. Whereas if you look at their spring peas, generally quite low, comparatively. So actually those winter peas were out yielding them. And then no response on the irrigated ones, which is odd. I'm not sure why that was. But in the dryland conditions, which is what we were mostly in, the winter peas actually did better. And this is Scotts Bluff. They only had irrigated as well. A couple things to point out. So this is the yield here, quite strong yields, 60, 70 bushels. We don't see that very often. Again, this is irrigated, so that's something to keep in mind. But the thing that I wanted to bring up from this trial, aside from the fact that it survived in Scotts Bluff, was that they did two tests. One was planted on September 16th, and one that was planted on September 29th of this particular year. And so they got about a 6 bushel difference. And the planting date itself is still, I think, an open question. We tended to plant ours a bit later, even into October, whereas our spring wheat window, we're generally shooting for about the 10th to the end of September. We want to be done with, excuse me, winter wheat. We want to be done with winter wheat by the last of September. And then we moved on to winter peas. And our logic was, as opposed to winter wheat, we wanted very little top growth of those winter peas that would be susceptible to some of that frost and some of that dieback. And so that was kind of the idea for pushing it a little bit later, and that seemed to work okay, but we didn't have a comparative earlier one. And so I can't say that that was the right thinking. That was just the logic, but it's not fully supported by the data. These guys, you know, it appears that they're a little bit south of us, but it appears that they had some benefit by planting a little bit earlier. Okay, so now I'm going to transition a little bit and talk about one of the reasons why I think peas are so beneficial in rotations. So we didn't really talk about that. I just talked about the market. But from a systems perspective and from an environmental perspective, peas offer a lot of advantages and really make a great rotational crop to expand our somewhat limited rotations across the Midwest. And so certainly the biggest or one of the biggest advantages of adding peas into the rotation is that they fix their own nitrogen, meaning you know, we have roughly 78% nitrogen in the atmosphere that can't be used by plants, most plants. Legumes have this ability to do that. They form this symbiotic relationship with microbes called rhizobia, who can break that strong triple bond that makes the nitrogen unavailable in the atmosphere. They break that and send it up to the plant through the roots. And in return, the plants give them carbon as energy. And so it's this kind of nice little back and forth that they have. And so a lot of my work, as I kind of started out this talk, I mentioned that we focus a lot on rhizobia and kind of trying to figure out ways to supercharge nitrogen fixation. Because right now I think the market is underdeveloped. I think there's some interesting stuff, but I think there's lots of room to grow. And so how can we make this system more efficient? How can we produce more nitrogen? Because ultimately that nitrogen that the legumes fix, you don't have to add it to that particular crop. So you can cross that off of your fertilizer bill. But B, some of that, not all of it, but some of that nitrogen will become available to future plants, either as the plant decays or through the roots. And so potentially we can have a lot of nitrogen savings with adding these. And so it's always one of the reasons why I'm such a big proponent of these plants, provided there are markets that one can sell to. So this slide is just kind of highlighting what I just said, that symbiotic relationship. And I'm going to go into this in much more depth here. I put this in here, and I wanted to kind of highlight the alternative and just show like how from just a physical perspective, how amazing this process actually is, what's happening in these little tiny nodules within the root. So the Haber-Bosch process, these two scientists, German scientists, won the Nobel Prize for figuring out how to do that fixation, that nitrogen fixation on a commercial scale, or how to do it on an industrial scale. And basically what it boils down to is it takes a monumental amount of energy. It takes obviously it takes the nitrogen and it takes some specific metal enzymes to make this process work. And so it's now known as the Haber-Bosch process. And that's where the vast majority of all of our synthetic nitrogen comes from globally. And so I put in here, so it takes 18 gigajoules of energy. So that doesn't really mean a whole lot to most people, I don't think, to produce a ton of nitrogen. Now, globally, 160 million tons of nitrogen is produced through this process. So that's 160 million tons times 18 gigajoules would be the total energy that it requires to produce this amount. for every ton of nitrogen, that energy, so for 18 gigajoules could produce or could power an average home for almost half a year, an average American home for half a year. So the total energy used to make nitrogen globally could power roughly 80 million homes for the entire year. That's just based on these numbers. And so that, and the amazing thing is that we're doing roughly this within the plants themselves, roughly this amount on a global scale is also being produced within the plants themselves, requiring only the energy from the sun. And so I wanted to highlight how much energy it actually takes to do this, and B, how amazing this process is from an evolutionary standpoint. And then in the process, it also emits quite a bit of CO2 each year to make all that nitrogen. So, okay, so these are the little nodules right here. These are, this is from field pea plants. And so these little weird looking things down here are the actual nodules themselves. And as I said, legumes in general, that family of plants obtains most of their nitrogen from the atmosphere, but there's this huge range. Field peas are generally pretty strong. They take a lot of their nitrogen from the atmosphere, whereas beans, common beans, are very poor fixers. Something like soybeans is kind of in the middle, and we've seen some trend with soybeans, in fact, that where nitrogen fixation has not been prioritized in the breeding process, and so it's not as strong as it could be. But I have here roughly 40% to 70% of the nitrogen within the plant generally comes from the atmosphere, and then whatever the difference is comes from the soil. But there's a huge range, 5 to 90%. There's about 5 to 20 genes responsible for nitrogen fixation, so it can be a pretty complicated process from a cellular level. Typically, there's 3 types of commercial inoculants. There's liquid, peat, and granular. I think liquid and granular are the 2 that are preferred. My preferred choice is granular. I think it does a much better job. I'll talk a little bit more about that. It's a little bit easier to handle, and so I tend to prefer the granular. Nodulation should be visible by 6 to 8 weeks after planting. So it takes a little bit of time for that to get started. But once the roots finally develop and they establish this symbiotic process with the rhizobia, then you can start to monitor that. And generally, nitrogen is not recommended with these plants. And in fact, it's discouraged in most cases, because the more nitrogen you get, the less nitrogen, the more nitrogen that's in the soil, the less fixation that typically occurs. And so, the plant tends to get lazy, we say. And so it's actually easier for it to take up that nitrogen from the soil than it is to give all of its carbon to the rhizobia. So if it's there, it'll take that nitrogen. But then in that case, you're leaving money on the table. So this is just another one. This is just a bunch of different legumes, and some of the amount of nitrogen they fix from the atmosphere, or as a percentage, and then the total. This is in kilograms per hectare. The conversion is about 90% of that for pounds per acre. So you can see there's quite a range. But at the high end, you could get over 200 pounds of nitrogen. Faba beans in particular, quite high. But depending on conditions, it can be quite low as well, or almost non-existent, which if there's time, I'll talk about. This is just a little bit of data from Canada. This is showing the yield on the y-axis here in different types of inoculant formulation. So this is the uninoculated control, so nothing. All they're relying on is the rhizobia that's native within the soil. And then you have liquid, peat, and granular, and obviously granular did the best in these trials by quite a bit. We don't see, to be honest, I've never seen this dramatic of a difference in all of our trials, but a lot of that I think has to do with how you handle the liquid. It's just, if it heats up, you can tend to kill off all the bacteria, whereas the granular is a little bit more protected. And so any heat, even if it's after you apply it, heat in the ground tends to kill the liquid rhizobia a little bit quicker, easier than in the granular applied stuff. So certainly some important differences, but I don't think the liquid is quite that dramatic if it's handled properly. This is just I put this in there. This is kind of an assessment if one was interested in looking at how vigorous the nitrogen fixation is happening, or how vigorous it's doing, how vigorous it is, I guess. And just to check, well, did my inoculant actually work? So I said that, typically 6 to 8 weeks or so, you can go out and check and see, are these nodules, you can see a little picture here, are these nodules present on the root system? And how do they look? So there's a couple different ways to score these. The first is just the growth and the vigor. How that's basically the plants. Do the plants look green? Do they look strong? Or are they chlorotic, meaning are they yellowing? Are they lighter color? And that may not occur if there's a lot of soil nitrogen. You might not see any color differences. The nodule color and the number, this is an important part, maybe the most important diagnostic part. So typically, as I showed this picture back here, well, there's this picture, there's a red color, same with this one. And that red color is critical. So within the nodule itself, there is an enzyme that the rhizobia uses to break that triple bond in the nitrogen, the N2. And that enzyme, called nitrogenase, is deactivated by oxygen. And so the plant, or this nodule environment, this system has developed to use leg hemoglobin, which is similar to hemoglobin in blood. It's an iron containing protein or compound that binds that oxygen and moves it out of the system. So it basically keeps the oxygen away from that nitrogenase enzyme. And that's the same thing that basically that the iron or the hemoglobin in our blood is doing. It's shuttling oxygen through our body and our vascular system. So it's the same process. And so the way that you can tell that this is these nodules are actively fixing that nitrogen is that you have that nice red pinkish color that denotes that process. And so sometimes you'll see these white nodules, which are just basically young, immature nodules that haven't started fixing nitrogen. Sometimes you see green nodules. Typically, green nodules suggest some sort of stress, like if it's from both ends of the water spectrum. If it's waterlogged or if it's drought, you get these greenish nodules, and they're just not really actively fixing nitrogen. And then you'll see black nodules, which are basically dead. Brown or black nodules that are dying or dead. So that's like once it goes through the green phase, then it goes to the brown or the green, or excuse me, the black. And then where they're located, they place some priorities. So on the crown of the plant, that's typically where you see most of the nodules, because that's where, as you know, those roots start to elongate out of the seed, that's where your inoculant was on that seed. And so it tends to colonize that crown area the most. And then it starts to spread with the roots. And now one thing that is one advantage we've seen with the granular inoculant is that you see much more diffuse. Obviously there's these strong crown nodules, but you see a much more diffuse nodulation forming on some of these feeder roots and some of these secondary roots. And generally that's thought these lateral roots have a lot more add to the total fixation process. Now the crown roots are probably the most important ones. That's where most of your nitrogen is getting fixed. But if you see a good distribution, that's generally what we're looking for. I'm going to skip this one. Running out of time here. Basically, rescue nitrogen applications are pretty difficult to do if your nodulation does fail. So what are some of the factors that are affecting nitrogen fixation? Photosynthesis is obviously one of the biggest things. So if the plant's not fixing carbon, if it's not growing, the nodules and the roots won't grow either. And for whatever reason, there's lots of reasons. There's too much water, there's too little water, there's too much heat, there's too little heat, pH, any of these things. And so if it's not fixing or if it's not fixing carbon through photosynthesis, then generally that's going to affect your nitrogen fixation. Because as I have here, 16 parts energy to nitrogen fix. So that's basically like 16 parts carbon to every 1 part of nitrogen fix. So it's a big expense. It's roughly 20 to 30% of all the carbon that's fixed by the plant is shunted down into those roots and taken up by that rhizobia. So energetically, it's expensive. The other thing is that that energy that's being used is ATP. It doesn't even triphosphate. So the P in there, I just said, is phosphate. That's pretty critical for both for root growth and for energy. So it's essential to have adequate phosphorus in the soil that's available to the plant for nitrogen fixation. Soil pH can be problematic at both high and low pHs. For one reason, phosphorus tends to bind pretty tightly at both high pH and low pH. It might be to calcium in high pH, might be to iron or aluminum in lower pHs. So you don't have the phosphorus availability, but you also have effects to the bacteria itself and some of the other nutrients that are needed. When you get out of that more neutral range, then you start to have a lot more limitations for a number of different nutrients. And then high clay content tends to decrease nitrogen fixation. Typically, that's because of water logging. So you get a lot of clay content tends to hold more water in its system for longer. And so you don't get that oxygen flow and you have a reduced rhizobial population. I'm going to go ahead and skip these here. Basically, what they're saying is long-term fertilization tends to produce lazier nitrogen, we found, or others have found. So now I want to talk really quickly about some of the work that we've been doing on rhizobia, specifically in response to some abiotic stresses like drought and salinity. And so I think I mentioned this earlier, that rhizobia occurs naturally in all soils or most soils. And so we don't really know much about it, but there's all these rhizobial species, and they could serve as this vast genetic resource for creating new rhizobia. Basically, we're breeding rhizobia similar to we breed plants. So what we've done over the last 4 years is collected soils from all over the plains, and we're looking for unique rhizobia species that have adapted to harsher climates. So, some of the dry areas like the Badlands in North Dakota and South Dakota, or some of the mountains in Montana, some of the grasslands, some of the prairies. So we gathered these soils from all over the plains, and we cultured hundreds of these rhizobia, and we started testing them in the lab for stress tolerance. And so what I'm showing here is some of those that we have produced, and we now move those. They did well in lab testing, so we moved them into some field tests. And so very quickly, I'm going to run through these slides. So these are box plots again. What I'm showing here is biomass. So this is the field P biomass on the y-axis, and these are different rhizobia species. So we have these numbers don't really mean anything of value. These are just native species that we cultured and that we think are tolerant to abiotic stress. This is a commercial granular, a commercial liquid, and then an uninoculated control. And then the red and the blue. So we planted them at 2 times. One is late May or mid-May. One is on time, which is mid-April. So roughly 3 to 4 weeks apart. So this red again is the first planting. You can see the biomass in general is much higher across all the reds than it is in the late planting. And I showed the similar data already on that, why that happens. You can see in 2021, much lower overall differences. 2022, much better growing year. So you have much higher total yields and much greater separation between the on time and the late planting. And so the nice thing we saw is a pretty decent trend from some of our native species versus these commercials. We see some increases in overall biomass production. Now this is nitrogen fixation. So this is in percent. This is how much of the nitrogen within the plant is derived from the atmosphere in these 2021 and 2022. And I didn't say these are in pure location. So same native species, same planting scheme. Generally, you know, we had roughly 50 between, I don't know, 55, 50 and 58% of the nitrogen in this really drought heavy year. And we see this maintained. So the nice thing, what we're trying to see is that our native species are higher than our some of these commercial products. And so even when it's stressed, it's still fixing more nitrogen. And that's true, but you see there's these massive data ranges. So it's changing within its own species. The variability is extremely high. And for me, it's very frustrating to do statistics on these. But we see some generally positive trends where some of these are fixing more of the nitrogen is coming from the plant than our commercial ones. And you can see with the uninoculated, it really falls off for this trial here. The amount when you don't inoculate at all, how much lower the total nitrogen fixed is in the plant, meaning that the native species that was here in this system is a much less efficient nitrogen fixer than some of those that we've selected. And I realize I'm moving through this really quickly. I apologize, it's just lack of time, but this is the total nitrogen fixation. 2021, very poor year. These differences, there's some differences between our native species and our non-native species. Some of it's not very great, but if you look, this is only 5 to 10 pounds difference. So 2021 was a poor year overall. 2022 was a much better year. And in fact, our best species fixed about 110 pounds of nitrogen per acre, whereas our uninoculated control at the on time was quite low, 60, 70 pounds. So there was, with these native species, there's the potential. I'm not saying this is guaranteed, but there is the potential that we have some fixers that do much better than even our controls, our commercial products, still fixing 10, 20 pounds more nitrogen. Then though, the unfortunate thing with this particular case is that when you go to the stress period, the late one, nitrogen fixation was not all that different. So we kind of inadvertently selected for these really strong nitrogen fixers, but really what we want to see is much higher total nitrogen fixation during these stress periods as well. And so that's kind of ongoing research that we're doing here. Very quickly, a couple minutes here. We also are looking at salinity. I just have 2 quick slides. Field peas are generally much more sensitive to salt soils in the soils than wheat is. You can see it's much more linear here, where this has a little bit of a threshold that hangs on a little bit more. We tested a lot of different rhizobia in the auger in the lab. And these are just basically, these colors mean increasing salinity levels. And so like, for example, this one did really well when there's no salt, but when you add salt, the growth just dies. This y-axis is rhizobia growth in the lab. What we're looking for is something like this, where it maintains growth even during higher salt levels. And you can see there's just a lot of variability when we start looking at them. We did this in testing in the field as well. We tested a couple different pea varieties, some that we thought were salt tolerant, some that are not. With some of our inoculations that we've created. And this is the test. We had one hailed out, so we had to put this in the fall. Basically high EC. This is saline soils, high sodium, and very high pH. So these are really rough soils that we were testing in. And the take-home message really is we see also this interaction with genotypes or with the peas themselves. And so some of like this is one of our native species here. It responds much more strongly to this particular field pea variety, whereas it was much more similar to the others. This is total biomass, total nitrogen fixed by the atmosphere. Now you're looking at the scale here. In these really salty conditions, we're only fixing somewhere between 20 and 40% of the nitrogen, and the plant is from that, from nitrogen fixation. And very low levels of overall, like this one, we saw great response, but still we're talking 5 to 15 pounds of nitrogen fixed. So this scenario would be probably like a cover crop in the fall. You know, it was planted in late August and harvested in late October. So, you know, we only fixed, we fixed quite little nitrogen, but it's something we're still working on it. What that means from a producer standpoint, especially from a cover crop in this case, is that there is some potential to grow. I mean, this blue and this yellow, this is blue is uninoculated, yellow is a commercial, or excuse me, other way around. This is a commercial, this is an inoculated one. We still get some growth, and so in these saline areas, even in the fall as a cover crop, there might be some potential to fix some nitrogen and to maybe try to reinvigorate some of those systems. That's one of the other things we're working on. We do a lot of DIY where we try to get make these things as simple as possible with a lot of homemade stuff. Kind of run out of time to do that, but I'm going to skip that. So I'm going to close. I apologize for rushing there at the end, but just wanted to thank SARE, USDA SARE, for their support in this research, and also South Dakota pulse growers, and of course all of the lab members, grad students, and postdocs that worked on these projects.